Well, with that, I want to turn things over to Frank Rulli from Institute of Evolutionary Medicine at University of Zurich. And he's going to introduce our speaker today, Verena. Go ahead, Frank.
Okay, thanks, Charlie. Thanks, Meredith, for organizing everything. It's my great pleasure, and have a nice evening from Europe. Actually, we have sunny weather here, so it's great. It's my great pleasure to introduce Verena. And you know, when Charlie and Verena and myself, you sort of discussed the exact date, I was not aware of the fact that we today actually have the Independence Day of France, so the 14 Juillet. And I think it makes sense because we are talking about revolutions. We are not talking about only the French Revolution today, but also about the revolution in terms of science. And I think Verena is a good example sort of highlighting this revolution. Verena actually has a diploma in biochemistry, and then she has a PhD in biochemistry in 2010. She got a PhD in scientific archaeology in 2014. 2015 to 2017, she was a group leader for paleogenetics at the University of Tübingen in Germany. And since roughly 2.5 years, she actually joined our institute as an assistant professor and head of the paleogenetics groups at the University of Zurich, where she is a member of the medical faculty and also of the science faculty, so a dual professorship. And we are honestly very happy to have you here. She started building up our lab, our ancient DNA lab, enlarging it, and I think she was able to get some very nice scientific results in the last few months, years, and she will hopefully talk about these. And so I hand over to Verena. It's a great pleasure to have you here. And again, as Charlie said, keep your questions for the end, and then we are happy and to answer them. And I'm pretty sure there are a lot of questions, because not only because it's a revolutionary topic, but it's, as you all know, in the last few months, it's a topic which is highly relevant. You know, talking about genetic changes of particular pathogens is something which we all are fully aware now in these days. So thanks everybody for joining, and I hand over to Verena.
Thanks a lot, Frank, for this nice introduction. And of course, thanks also a lot to Charlie and Meredith for having me as a speaker. It's a great honor to be here. And last time also Charlie mentioned that it will be the first ancient DNA talk in the club EvMed. So well, I hope that I can also introduce a bit of ancient DNA to you, basically show you what actually is the potential and how we can basically use that to understand pathogens much better. And so I will now start sharing my slides in case that it doesn't work. Let me know. So here we are. I hope you can all see my slides now. Should be with a starting slide. Okay, then I basically wanna invite you in on a small time travel, and we basically will go back to early modern Europe later on and see what Treponema pallidum basically is doing there. But before I basically want to start a bit with the with ancient DNA and also with the concepts on how you can actually look at ancient pathogens and how you can basically study them in general. And well, you probably are aware of the current pandemic, but that's not a new thing. So pathogens have been with us in human history for a very long time. And they also have an impact on us, either directly as human pathogens or as pathogens of our crops or animal livestock. And they can basically really change and also influence history a lot. If we look at historical pandemics, this is just a selection, so there are, of course, many more, but these are some of the very striking ones. You can already see a problem that is occurring with those pandemics. We find them in the historic textbooks, but we can usually not really trace back the causative agent. So if we, for example, look at the plague of Athens or Justinian plague or the Black Death, so there are a lot of causative agents discussed, and it's not really clear from the descriptions from the old medical textbooks and so on to actually reconstruct it and see what causative agents there might be. And this is actually one thing that we can contribute. We can actually identify causative agents of those historic pandemics and basically use them to study long-term evolution of pathogens. So we can basically see evolution in action. We can travel back in time, look at the genetic signatures of those pathogens, and basically compare them to modern and see how this pathogen evolved over a very long time. And we can also, of course, use our molecular fossils to calculate mutation rates over a long time that are hopefully then more accurate than those that you just get from calculations in the laboratory. And you can also calculate most recent common ancestors with including those ancient genomes. Then, of course, the question is, how do we do that? Well, it's probably obvious you need ancient DNA for that. And ancient DNA is, well, a bit different to modern DNA. And you have basically, if you want to work with ancient DNA, you have at least 1,000 times less DNA per gram of tissue than you would have. You also have it fragmented. So the DNA we deal with is usually between ancient DNA is also chemically modified. So this usually comes from the DNA aging in the soil. So we have here you can see, I hope you can see my cursor, but here you can see a generous trend of the DNA. And you can see with all those little arrows, you can see what all can happen to this trend, to the DNA while it's basically stored in the soil, and all those chemical modifications and all those chemical reactions that can take place. And they basically lead to a characteristic damage pattern that we can also later on detect in the ancient DNA. This is, of course, problematic if you later on want to analyze it, as you need to somehow distinguish those damage patterns from real mutations that you want to look at later on. However, they are not just bad. You can also use them to show the authenticity of ancient DNA. And if we have such a typical ancient DNA strand with those overhanging ends, such a fragment here with those overhanging single-stranded ends, then these ones are the most likely sites for DNA damage. And we can basically detect that. And this is this characteristic C2T exchange, which we also see here as a characteristic smiley pattern. So here you have a fragment plot from the inside of a fragment to the outside. And you can also see that for ancient DNA, we basically get such a smiley plot, while for modern DNA or for contamination, we don't see that. So such a damage profile is not just a problem for analysis, but it's also important to have that to show that you're dealing with real, authentic ancient DNA. So if we are now coming back to an overview of the problems when you want to work with ancient DNA, we have this very low amount of template, but for this we have developed a lot of different extraction methods that actually work very well and allow us to deal with that. Then we also have some fragmented DNA, which is around usually 50 base pair long. But here, luckily, the next generation sequencing is very helpful. So with Illumina, we can sequence millions of short reads. So that's actually nice for us. And then, of course, a question about the contamination. Well, over there, you can actually see me in the clean lab working. And this might remind you a bit on the current pandemic. And we also wear Tyvek, face shield, mouth protection, and two layers of gloves. However, we are not so much afraid of getting any virus or catching any virus. We basically do it to protect not us, but the DNA that we are working with. So we basically wear it for a complete opposite purpose. So we basically try to limit our contamination that we might bring in and basically not contaminate our ancient DNA. And then we have the last point is, of course, the chemical modification. This is what we have learned already, that it's not such a problem. There are ways of later on dealing with it, and either in the lab or bioinformatically, but we need it also to show that we really work with ancient authentic DNA. Sources for ancient DNA, well, we are very flexible. We work with a lot of things, mainly teeth and bone, but also soft tissue, dental calculus or coprolites. And if you are interested in working with plants, well, seeds from archaeological remains or herbaria also work very well. And there are even applications on retrieving ancient DNA from soil samples. However, we of course have limitations, so the DNA is degrading over time and it's getting less and less. So the calculations about the maximum age for DNA retrieval is around 1 million years. So things that are older are not basically not possible. So we can actually, unfortunately, forget about our Jurassic Park or Jurassic World. But if there are any people interested basically in those parks, well, we can actually help out with nice Pleistocenic animals like mammoths or cave bears or saber-toothed tigers. So for these we have already, there are a lot of publications around from various groups and various teams, and they have already sequenced genomes from those animals and are also specialized on that. The oldest so far sequenced genome actually comes from Ludovic Orlando's team. It's a 700,000-year-old horse genome. So, but now coming back to pathogens and ancient pathogens. So the problem with the pathogen is that we basically do not really have fossils from pathogens. So therefore, we always have to go back to the human host or to the pathogen host in general, and it gets the DNA from there. And if you look at a characteristic human tooth, then we basically do not only find human and pathogen DNA in it, if we are lucky, but also a lot of environmental contaminants like plants or fungi or bacteria that actually lived on the bone, left their DNA behind and create a huge environmental background that basically is problematic for us to later on sequence our pathogen DNA. And therefore, we always do so-called targeted enrichment for later basically fishing out our pathogen of interest and reducing that background. And this is a bit of our schematic workflow, how we do it. So if we have our tooth, then we basically cut it in half, sample it from inside of the enamel, and then basically perform the DNA extraction with basically obtaining human DNA, pathogen DNA, and environmental DNA. If we turn that into a DNA library by adding artificial adapters, then that percentage stays the same. But if we do then the enrichment, where we basically fish out our DNA of interest and reduce the background. So the enrichment is basically, here it's a class, it's an array or an in-solution capture. The principle is always the same. You have basically artificial bait libraries that, artificial bait DNA that basically spans your genome of interest and fishes out all the fragments that you're interested in and basically reduces the background. And then you do a sequencing and bioinformatic analysis. So this was basically the short introduction on a bit of the methods and how we do it and how we deal with ancient pathogens. And now I basically want to show you the example I hope you're all curious about. And we basically start with our Treponema pallidum, so syphilis. And syphilis is actually caused by the subspecies Treponema pallidum pallidum, while we have other Treponema pallidum subspecies that are causing yaws of Bachelor in humans. But in particular, syphilis is very famous and the first reported outbreak in Europe was basically taking place at the end of the 15th century. And over there, you can also see the first depiction of a person suffering from syphilis by Albrecht Dürer. And ever since then, it has been one of the most feared and also stigmatized diseases. However, it's not a disease of the past. So be aware of that this disease is currently re-emerging globally and has around 10 million cases per year. And therefore, that's a very large health problem also for our time. If we now want to investigate a bit more about where it actually comes from, well, the origins are largely unknown and highly debated. So very quickly, there are two main hypotheses. One is the so-called New World hypothesis that is also supported from some modern data. It basically states that Christoph Columbus basically brought it from the New World to the Old World, and then there basically then had this first pandemic. And the multi-regional origin hypothesis basically states that it was already there and that some environmental factors probably caused this outbreak in the 15th century. And there we have some potential skeletal evidence, which is also highly debated. And you can already imagine what would be nice to have is, of course, ancient DNA to basically settle this controversy. And that is what I was working on for a very long time with some of my colleagues. And the problem is that ancient DNA and syphilis are not a perfect match because there was so skeletal lesions that are used to basically diagnose syphilis in the archaeological records are usually related to a very advanced stage. And there it's also problematic already in modern patients to detect the pathogen. So it's not an easy target. And therefore we basically tried to do that with using capture-based tools and NGS. And I quickly want to introduce you to our 2019 paper that I worked together with colleagues from the Max Planck in Jena and from Mexico City, where we sequenced the first Treponema pallidum genomes from colonial Mexico. And here you can see an overview of our positive samples. So they basically come from the convent of Santa Isabel. And that convent is actually quite interesting as there have been a lot of individuals buried, but they are mainly fetuses and neonates and infants. And we were actually able to look at 5 of those individuals in our study. And they all had signs of infections, which are characteristic for treponemal diseases. So this periosteal reactive bone formation and this particular expansion of the long bones. And 3 of them were actually positive. So we reconstructed 3 genomes from between 3-fold and 7-fold coverage, and also obtained damage patterns that actually fits nicely to the dates that we from this early modern period. If we now look at some interphylogeny, then you already can see the first striking and very interesting finding. We compared them to the modern genomes that are available and basically found that only 2 of them fall together with the syphilis-causing strains, while we have another one that basically falls together with the yaws-causing strains. In addition, we also found indications for putative recombinations. And I don't want to go into details, but I'm happy later on to discuss this, hopefully then when we have some time for discussion. So please ask about it. And we also, of course, looked at virulence factors. And interestingly, our ancient yaws strain looks completely the same as the modern ones. However, our ancient syphilis strains have one missing factor. And this is actually a homologue of a transport long chain fatty acids. However, how this really affects the virulence is not clear yet. So, but this is an interesting finding regarding the differences in virulence factors. And now we basically want to finally come to our new results that I hope you had a look at the preprint. So this is mainly the work of my postdoc, Kertu Marianda, and my PhD student, Saskia Prengle, and they all worked on Treponema pallidum in Europe. And we also got very lucky to obtain a grant from the Swiss National Foundation last year, so that enabled us to really work on that very hard and also concentrate on that. And we were able to obtain 4 genomes from Finland, Estonia, and the Netherlands. And this time we also had an adult individual among those among the victims. And so not just infants. It was also very interesting. And we have also a very high coverage genome, up to 100-fold coverage. And they are dated in the 15th and the 17th century. Now you might ask if some of them are pre-Columbian or not. Well, that's not so sure. We are not so sure about that, but we can later on discuss it more in detail. So we say it's contact period to be on the safe side. And if we now put those into the phylogeny, you can even see that we have some very interesting new findings. So 2 of those genomes actually fall with the syphilis causing strains. So those 2 guys here. And we have 1 that falls with the yaws causing strains over here. And 1 very interesting genome that forms a basal lineage that is basically basal to yaws and Bacil causing strains. So over here. Overall, we can find a very high diversity that we didn't expect, and at least 3 lineages of Treponema pallidum that are present in early modern Europe. We, of course, also did now finally some molecular clock dating on it. And you can see the posterior distributions of the divergence state for the different clades and the samples over here. And in the other picture, you can actually see the maximum clade credibility tree. And overall, what you can take from those are basically the different most recent common ancestors. And for the whole Treponema family, we basically have it for at least 2500 BCE. And for venereal syphilis, it's between the 10th and the 15th century. For yaws, it's actually later, so 15th to 16th century. And the divergence between yaws and Bacil is quite wide between the 9th century BCE and the 10th century CE. And of course, finally, last but not least, also the virulence analysis. Well, compared to our strains from colonial Mexico, which we have over here, where we can clearly see that this particular factor is absent, here we cannot say that. So for those, it seems that there is no difference in the virulence factors compared to modern strains. If we now summarize that, well, we have now those 4 ancient genomes from early modern Europe, and they are highly diverse. They include syphilis and yaws causing strains, as well as this unknown lineage. And we have also this very new lineage that is basically this is a basal sister group to yaws and Bacil causing strains. And the molecular clock dating per se does not exclude this pre-Columbian origin. So overall, let's say this diversity and history in the old world. And
I think Verena, you were frozen a bit.
Okay. So
maybe good for DNA preservation, but not for the Zoom meeting.
okay. So when should I start again?
Just a few sentences before.
Okay. Okay. Well, I hope you all heard still about that molecular clock dating that does not exclude a pre-Columbian origin. And in general, what we can take from that is, while there, together with the with our molecular clock dates for the different most recent common ancestors, as well as this very ancestral lineage we have, there is somehow a history of Treponema pallidum in the old world. We can, of course, not exclude that there were some new lineages coming in the 15th century, but in the moment, our data does not exclude, so it could also go in the direction that there might be at least partially an endemic origin of the 15th century pandemic. So this is basically, in the moment, well, we are still not sure about that, but at least we can say that we have some indications that at least Treponema pallidum was there, but we can only be sure if we have real pre-Columbian samples. But of course, it's a very interesting finding that we see that high diversity. And I'm happy to discuss this later with all of you, what you think about this high diversity. And I basically just want to close with our summary for the ancient pathogens. So I hope that I could convince you that it's a really interesting discipline, and we can really trace back pathogens over time, identify causative agents, and also basically get insights into the evolution of pathogens via this comparison of this ancient and modern strains, and basically also detect strains that we probably do not see today anymore. And we can also use those for calculations of mutation rates. And we can, what we haven't talked about yet, but I'm happy to discuss this as well, we can also use the developed methods for other fields, especially since we're dealing with very low DNA content that might be also interesting for other fields. And this is basically, these are my colleagues that contributed to the latest Treponema pallidum study, and my group in Zurich, as well as our funding bodies, especially the Swiss National Science Foundation study. And in the end, I also want to give you an outlook, and I've also seen
we lost you again.
Yeah. Okay.
That we
just lost you for a few sentences again.
Okay.
Basically for the whole slide.
Okay. So this is also my last slide, then I'm done. And I hope then that the university internet connection is a bit more stable than when I stop sharing it. This is my outlook and also my last slide. And this is, in case you want to know more about ancient pathogens, I'm co-editor together with Anne Stone, who's probably also participating in our meeting, and Cecil Lewis, as Royal Society special issue about the insights into health and disease from ancient biomolecules, and I hope that you all will have a look once it's out somewhere in September. So again, thanks everyone for listening, and now I'm really curious about the questions.
Thanks, Verena, for a great talk. I think you showed very nicely the methodological problems, but also the applications and obviously the impact, like, for example, the change in virulence and so on. And I think there's a lot to discuss. And we already have actually a few questions in the chat, which I will start raising. And I think once we have done the questions in the chat, we open the mic to everybody. As Charlie said in the chat box, please raise your virtual blue hand if you want to ask a question, but I will start with the 2 or 3 questions which are already in the chat box, if you don't mind. So the first one was, can you also retrieve pathogen DNA from the calculus on the teeth? And if so, what is the benefit of extracting pathogen DNA from inside the tooth?
Well, there are some possibilities for dental calculus. And but this is also particularly interesting for oral microbiome studies. So of course, there if you can retrieve, for example, red complex bacteria and like Tannerella or Treponema denticula. So this works very well for oral microbiome in general. Why we basically always go back to the inside of the tooth is because there it is, first of all, it's very well preserved. So it's like a little time capsule. And we also have there the best opportunity to catch bloodborne diseases like Yersinia pestis or others that there we can basically catch those best.
Thanks. I move on to one or two more questions from the chat. Like for example, someone asked, please elaborate why the pathogen cannot be easily detected at the advanced stage of the skeletal lesion, even in modern samples. I think that was related to treponemal disease.
Yeah, well, in this case, I can only rely on the clinical publications where they say that it's basically very hard in modern studies. If you have already a patient with advanced stages of syphilis, that they can only basically get antibodies or something like that, but it's not so easy to actually retrieve genomic information from the pathogen anymore. Why that is the case, I think that's not really studied, but it seems like that the pathogen somehow tries to escape the immune response or some in whatever direction. Basically, it's not really clear why this is the case, but it looks the other idea is, of course, that some of it is of the damage is also caused by the immune response of the body and not by the pathogen itself. But this is basically all due to clinical literature what we found. And well, we can see that at least with active lesions or also with congenital syphilis, we can actually still detect it in bone samples. Yeah, I think there's one hand, right?
Let's go for the live questions then. Okay, Brandon Ogbunu.
Hi, yes, can you hear me?
Yes.
Yeah, okay, great. Thank you. That's a really fascinating talk and a topic I'm only vaguely familiar with. So I think using the technology to kind of resolve a kind of an ancient human epidemic is a really, really important, interesting use of the technology. I can't help but think about, other animals where one might be able to kind of use and extract ancient DNA in order to kind of capture, for example, zoonotic shifts, for example. I mean, is this the type of thing that people are using this technology for?
Well, but you mean with ancient animals? So there, the problem is that they are not so often preserved, and this is basically in the moment still a very new field. And well, especially small animals like rats or other rodents, it would be very interesting to study. Especially in old excavations, they are not preserved, so they are really hard to catch in the archaeological record. And but of course this would be a very nice application. But I was talking about the applications in other fields are, for example, pathogens that cannot be cultivated in the lab, like Mycobacterium Mycobacterium leprae, or also syphilis. There you usually have, like for example, Mycobacterium leprae, there you usually have to passage it through animals if you want to isolate it from patients and then sequence it. And with our techniques, you can basically directly enrich it from patients and avoid that animal passage, which is actually quite nice.
Maybe related to that, we have a question saying that what about animal pandemics? For instance, the Tasmanian devil facial tumor seems to be a new disease, but is it? Would DNA tracing help define it and its origin?
Well, ancient DNA, well, DNA tracing in general maybe, But I think it's very complex, and we don't even know if it is a DNA virus, or so. If it is an RNA virus, then it might be really hard to basically find those in historic records, where we could actually look at, for example, old Tasmanian devil samples also from the historic time. But if you, it might be quite hard to actually find that. And I'm sure that a lot of, as I'm quite sure that a lot of specialists are basically working on that, and also using those enrichment techniques and looking basically for that. But yeah, as long as we don't really know what virus it is, it's probably very problematic.
We have quite a few questions about virulence and very specific questions, like, for example, does the design of the capture baits include TPR gene regions which are important in pathogenesis of Treponema pallidum? If so, how are those regions from the ancient samples affect the virulence analysis of your latest study?
Well, yeah, there are, of course, we have, of course, included them, and we have also looked at them. The problem is that they are very similar to each other, so it's sometimes very hard to distinguish to which of those TPR genes they map. So what we can only say basically that they are in general there, but some of them are, due to the short fragments, it's very hard to basically completely reconstruct those genes completely. What we can say, of course, we had a look at them, and we also basically found that some putative recombination events, and so of course we had to remove them later on for the phylogenetic and for the dating analysis, but they are in general recovered in our early modern genomes.
There are some questions about past pandemics, obviously. For example, could you comment on the controversy that the Black Death in Europe was not caused by bubonic plague, but may be by a virus, and what efforts are being made using ancient DNA to answer these questions?
Well, from the ancient DNA side, it's very clear that it was actually caused by bubonic plague, as we have found this in a lot of plague cemeteries. That was actually, I was also involved in the first, the reconstructions of first genome from the Black Death, and I have lots of colleagues from the Max Planck Institute in Jena that are basically working on that, and there are several studies on that. And from my point of view, I'm very convinced that it was caused by the bubonic plague.
Charlie wants to ask something live.
Oh, there's also Kelly who also has a hand up.
Kelly as well, yeah. Whoever goes first, I don't mind.
Okay, well, I was just wondering about your thoughts on the evolution of virulence in the historical record. I know some people have proposed that there's some evidence that virulence of syphilis was very high in the late 15th century, and it was proposed that that was it was related to its recent introduction to Europe. And I was wondering if you had any thoughts about that historical record, and if you could help orient us toward how your results might bear on that. It sounds like you don't think that there's any evidence of virulence evolution, but I wasn't totally clear on that with the virulence results.
Well, in the main issue that we have to face actually is that a lot of virulence factors, we don't really know yet how they are completely linked to virulence. Especially in syphilis, they are understudied. For our early modern genomes from basically our ancient genomes from Europe, we can see that they have the same virulence factors than the modern strains, basically from our historical European genomes. There we can basically see that they have them covered. for the colonial Mexico genomes, there we see that there's one missing. However, we cannot really tell from the literature how, if there's any effect from that homologue arising regarding the virulence. So that's, I think that's a main issue, and I hope that colleagues from the modern treponema fields are investigating on that and help us out and to find out more about this particular virulence factor.
Thanks.
There's also a question about sort of an analog to, oh, we have Kelly Blevins, sorry. Yep.
Oh, no worries. Hi, Verena, great talk. Thank you. Exciting results. So I have a question about the recombination that you detected. So recombination affects mutation rates. So I was wondering how you dealt with that in your dating analyses.
Well, I can actually quickly share with you slides regarding the recombination analysis. Well, actually, this is mainly the work of our collaborators, Fernando Gonzalez and his PhD student, Marta. And what we basically do, we identify those putative recombinant regions, and then we remove them for dating and also for the phylogenetics analysis. So this is basically what we detect for certain for the different strains. I can also stop sharing now, and so this is basically these are some of the recombination events that we are detecting, and of the putative ones, and all of those are removed for the phylogenetics as well as for the dating. And if you are a bit more conservative, even then you need to remove even more, and then you get even basically more precise intervals for the dating, for the molecular clock dating. But yeah, that's a general thing that is also done in modern treponema research, that you basically always do all those recombination analysis and then basically takes this out, as otherwise you would basically need to do networks to really detect that. We have also done those networks. Unfortunately, I didn't bring them, but they are also really fascinating. So that's the thing actually to investigate on a bit more on recombination in general, as this is, as you basically take out a lot of information just because you're basically removing all those recombinant, putative recombinant elements and so on.
I think we have Ron Willis who wants to ask a question.
Unmute. Thanks. Given the samples from Estonia and Finland, might it be possible, is there any evidence that Leif Erikson and the Norsemen brought it back 1,000 years ago from North America?
Well, in the moment, unfortunately, that's a very interesting question, but we don't have any evidence for that. But yeah, we don't really know. Let's say in the moment, it looks more like that at least one of those subspecies could be already around in Europe. And about the other ones, we of course don't know how it came in, but at least around the contact period, we have all those 3 lineages. And that's very fascinating. But how the Vikings also might be involved, we don't know. But there are, of course, a lot of other trade possibilities to Africa and Asia. All those things could come to Europe. They don't always have to, they don't only have to come from the Americas. They could also be introduced from other countries, or basically already be partially endemic in the old world. So that's a very fascinating question, and I think we can only solve them if we have some pre-Columbian samples.
I think Thomas Strusacker was raising his hand. I don't know if you want to ask a question.
Yes, thank you. Yes, can you hear me okay?
Sure.
Good.
Yeah, okay. Yes, I'm interested in related treponema in yaws in particular. It's turning up now in a number of non-human primates in Africa. And I wonder if by going back to old collections, you could gain some idea of the history of the incidence of yaws in some of these animals. I mean, collections that were made, say, a couple of hundred years ago. And then also see how this might relate to the possible exchange with humans. You know, a lot of people in Africa will eat non-human primates. And sort of related to this, can you pick up treponema from feces? I mean, can you detect it in the feces? In other words, we'd like to know how far this is. Some of the animals are showing symptoms, some may not be showing, and of course, some can actually heal over with yaws, so you can hardly see that they've been infected, except you notice that they're missing a nose or something like that.
So far, I think there are some ongoing trials to do so, but so far the publications are usually around blood samples, and also there are there's one publication already quite a while ago from recent bone samples, but let's say in the moment it's all about recent animals, and they're
Pause again.
the ones that are found in humans, and therefore
we're losing you.
Verena, you're frozen again.
Yeah. Skype
has some problems in the moment.
Yeah, you have frozen again.
Yeah, hope you can hear me again. So those are usually based on those publications are on modern samples, and so far it's usually blood samples, and it could be, of course, possible to get it from feces, but it's not really done yet, I think, from what I know. And there have been one publication on recent bone material, but it's always a bit problematic with basically reconstructing it from those material. And in general, it would be, of course, a really cool idea to go back and see what we can find in zoological records and see what if the primates have been infected for a long time period already. In a moment, it seems from the modern samples that usually all those publications basically find that the strains that are in the non-human primates are very similar to the ones in humans. So there have been some exchange probably.
Sure. Yeah. Thank you.
Thanks. I think we are slowly heading towards the end of today's Journal Club, but we still have one last question by the president, actually, of the International Society for Evolution, Medicine and Public Health, Barbara Nattison-Horowitz. Please, Bob.
Hi, thank you. That was a wonderful lecture, and so thought-provoking. I have one question. I've been curious, over the past decade or so, there have been reports of permafrost thaw, related release of some of these DNA megaviruses and spore-based pathogens, the anthrax, the boys who contracted anthrax in the Soviet Union. So I'm just curious if you could comment on that. I've heard that those viruses really get degraded and are not that usable, but I'd love to be enlightened about that field.
Well, that's a bit different to what we are doing. So our viruses or our pathogens in general are usually in very small, in very small fragments. So there's no possibility that they can infect people again. So they are basically dead, and they're highly fragmented DNA. Regarding spore forming pathogens that are frozen or so, that might be different in the permafrost if they are basically frozen for a very long time, but I haven't seen that they are really basically, that they have really tried reviving them so far. So this is a field that say usually we work with really bad pathogens, And we're also happy about that because otherwise we would need.
Oh dear, again. Oh.
Okay, we have lost Verena in the last 2 or 3 sentences.
Sorry again. It's like it's a university the connection. I'm sitting in my office in the moment, and it looks like that there are too many Zoom meetings ongoing in the moment. Sorry for that. So what I wanted to comment on is like we usually, as I said, we work with pathogens that are really dead, and ours are very fragmented, and we're also happy about that. We have very fragmented, highly degraded DNA, and we are happy about that because otherwise we would need a safety level for laboratory to actually be able to do so, to work with them. So in our clean lab, of course, we are in a way happy that our pathogens are not able to infect anyone anymore.
So that's a very positive note at the end. I think this is really the end of today's presentation. I would like to thank Verena again for her presentation. I tried to stay now in the main camera because I was looking at the other screen all the time, at the big screen. So it was a pleasure to have you here. Thanks, Verena. I also would like to thank again Charlie and Meredith for all the organizational stuff. I think the chat is going to be saved. So I think Verena is going to be happy to answer all your questions, or if you want to drop her an email later on, please feel free to do so. I think it's a great opportunity to sort of focus on a different perspective within evolutionary medicine. And this is it from my end. And I hand over to Charlie.
Okay. I'll also add my thanks, Verena. That was a great talk. It was perfect for Club EvMed. So thank you very much for joining us today and sharing your results and giving us some background so that we can understand this field better. And thank you, of course, Meredith, for helping to organize, and Frank for doing a great job moderating. And thank all of you for attending today. It's really great to see so many people joining us for these Club EvMeds. We have one more coming up next Monday. We have Cynthia Bell, who will be talking about hemoglobin and high altitude hypoxia in relation to evolution and public health. And so I think that'll be a very fascinating talk. And I know Cynthia is on with us today. So thank you, Cynthia, for joining us next week. We're looking forward to it. We're going to then take a couple weeks off. We're going to have a little bit of a break at the end of July and early August. And we'll start up again in about the middle of August. I want to call everyone's attention to an announcement we're going to make over the listserv, which is going to call for talks, short talks by finishing PhD students or early postdocs who might want to give a 10 or 12-minute presentation to share their findings. And we think that's especially important these days with all the cancellation of meetings and other things. And so we'll give you the opportunity to basically apply to give one of these talks, or for your advisors or other people to nominate you to give one of these talks. And we're going to have 2 or 3 sessions in the fall that'll be devoted to hearing from people who are just finishing up their PhDs or who are early stage postdocs. So stay tuned for that. Okay, thank you everyone for joining us today, and we look forward to seeing you again next week. Take care.
Thank you.
Bye.
Bye-bye.